Hello everyone, good evening. You were asking some queries regarding the exam and the notes. You can ask those queries in the discussion form. The T of this group is of this course. Maybe he will reply on the discussion form. So we will start the session. So I welcome you all in this problem solving session of the course named Cell Culture Technologies, and this is a week 3 session. My name is Ubaid Tariq, and I'm a PMRF scholar at BSB Department of IIT Kanpur, and I will be taking these sessions every Tuesday at 7 p.m. So we will directly start with the questions, and after that, I will brief you uh, about some techniques that are in the questions. So first question, you all can answer in the chat box. First question is, what is T20 in T25 T20 flask? What is 20 in T20 flask? Length of the flask top, height of the flask side, area of the flask base, volume of the entire flask. So most of you have answered it correctly. So the answer is area of the flask base. Like here in this slide, I have shown that there are different types of flasks based on the this area on which the cells are grown. So usually in cell culture there are T25, T75, T125 and T225 flasks. And these T25 flask has a base area where the cells are grown and on which the media is present. The surface area is 25 centimeters square. And same like in T75, the surface area on which the cells grow, actual surface area on which the cells adhere is 75 centimeters square. That's why they are named as T25, T75. And T means tissue culture flask. Second question, based on question 1, the dimension 20 into 20 is a meter, millimeter, centimeter square, cubic, decimeter. As I already told you in the previous question that it is the centimeter square. As most of the flasks, their area is in the range of centimeters. So the the dimensions they are kept in the centimeter square. What centimeter square means that I think I cannot draw here. So I cannot draw here, but you can see this pointer. So that means this 1 centimeter. If we divide the this base of the flask into the square centimeter, there will be 21 centimeter squares on the flask. So it is like that. Third question: to ensure carbon dioxide exchange in the T20 flask, which of these things are done? Close it tightly while in incubator. Leave it slightly open while in incubator. Leave it fully open in incubator. None of the above. Yes, fewer you are answering correctly. Leave it slightly open while in incubator. Can anyone write in the chat box or unmute yourself? What is the disadvantage of these types of flasks in which we need to open the this open the flask this uh, slightly open the flask so that the oxygen CO2 exchange will occur. The lid we have to unscrew that lid slightly so that the CO2 exchange occurs. But what is the disadvantage of these types of plasma? What is the disadvantage of opening this cork, opening the lid or the knob that is present? No one? So there are, yes, someone has answered that we have to be careful while taking the flask out. Yes, that is the one reason maybe. It may cause the contamination. There are two types of flasks. One flask is this non-vented flask, which does not have the filter or pores in this lid, and one other is in which there is a vent in the lid. So both have the purpose to use. For example, if you want to transport the cells from one place to another place, so in that case, the flask which has a closed this non-vented lid that is required. But usually in the cell cultures nowadays, the cell culture flasks for for placing them in the incubator. Usually these flasks with the vented tops they are purchased. These vented tops they have the filter of 0.22 micrometer. In this 0.22 micrometer only the CO2 passes through these filters. Only gases pass, but no microbes will not pass from outside to inside of the flask. But if some lab uses these types of flasks to grow the cells in the cell culture, so uh, they have to open this lid so that the CO2 exchange occurs from between the flask and outside of the incubator. So in that case, there are the chances of contamination, maybe because there is no filter when you open the this. So when you open the cap, so there is no filter. So CO2 will also go. Maybe microbes or contamination will also go into the flask, and it will contaminate your cells. While these have the micro 0.22 micrometer filters, and most of the microbes they have the size greater than 0.22 micrometer, and they will not pass through this filter, so it will prevent the contamination. But when you want to purchase the cells, or you want to transport the cells from one lab to another lab, or one place to another place, these flasks are needed, which have these non-vented uh, caps. In India, can anyone tell in India where from which is the laboratory or institute from which cells are ordered? You can order the cells. Yes, NCCS Pune. So if anyone have received the cells from NCCS Pune, those flasks have these caps. They do not have the vent, and in that case the media is also filled. This whole flask is filled so that the even if the flasks is upside down during the transportation, the cells will be in contact with the media. So in that case the whole flask is filled. Fourth question is, matricial coating is done prior to cell plating. What is matricial? Cleaning agent, extracellular matrix, antibacterial coating, cell suspension. Most of you have answered it correctly. So Matrigel, it's actually a trade name. Matrigel is a trade name. The company which synthesized this Matrigel, Corning Life Sciences, the name of the company. So it's actually the basement membrane matrix. It's the extracellular matrix which is isolated from the basement membrane of the mouse sarcoma cells. So it usually contains lignin, collagen 4. They are mostly abundantly present in this matrigel. So this matrigel, it is a substrate for culturing the cells. So as you know that the adherent cells, they do not adhere to the surfaces like plastic or glass. They don't adhere well, so then they need some material that should be hydrophilic also, and they should be there. That extracellular matrix also gives the simulations to our physiological system. So usually these matrigel, they are used for the culture of the endothelial cells. For example, in our body, the endothelial cells usually grow on the basement membrane. Endothelial cells grow on the basement membrane. So if we want to grow the endothelial cells in the cell culture, this matrigel is used as a coating in the cell culture plates, and this coating helps or gives the microenvironment for the endothelial cells. Microenvironment which simulates the environment of our body, which simulates the environment of where endothelial cells actually grow. So it has been shown that if the endothelial cells are grown without this matrigel on other coatings, for example, if the flask is coated by other extracellular matrix like collagen or polylysine, if it is coated by these materials, so the cells will grow, but they will not form the natural structure like spider web like structure. They will not form. So as you know that endothelial cells are usually mostly present on the blood vessels, interior of the blood vessels. So they will not form these structures. So these matrigel provides the exact environment of the physiology where actually these endothelial cells are grown. So using the matrigel in the cell culture, some of the assays have been fabricated, or some of the assays are done. First assay is the tube formation assay. As these endothelial cells, they line the blood vessels, and also the capillary is only made up of endothelial cells. So in that case, if someone wants to study the angiogenesis means neovascularization, formation of blood vessels. For example, some signaling molecules are added to the endothelial cells, and these endothelial cells have the property to make the new capillaries, to new blood vessels. So these signaling molecules give some signals so that these endothelial cells are formed. And this matrigel, it is also known that this matrigel also has the property that it provides some factors. As it is the extracellular matrix, it is actually the basement membrane, which is it is taken from the basement membrane extracellular matrix. Then it is fabricated in a gel type manner in a solution. After that, in a solution, when we coat the cell culture plates and place them at 37 degrees centigrade, this matrigel will polymerize and coat this plate. After that, if we see endothelial cells on this matrigel, after that time these endothelial cells will adhere to this matrigel. But if we provide some factors like vascular endothelial growth factor or some drugs, some novel drugs which have the property to increase the angiogenesis, to increase the new blood vessel formation. So when these molecules are added in the matrigel, the tube formation occurs like this. These endothelial cells arrange themselves in such a way that they form the tube-like structures, and this shows that these endothelial this is the experiment endothelial cell angiogenesis assay, where we show that for any molecule, for any potential molecule, if it has angiogenic potential or not. Another application of this matrigel, that it is used in organoid formation. As this matrigel, it is a hydrogel, and it also has the pores inside this. So if this matrigel is poured in a culture plate with the suspended cells, cells are suspended in this matrigel and they are poured, and after that, when this matrigel is placed at 37 degree, so it polymerizes this. So here the doom is formed. So this matrigel is poured at a particular place on the cell, and this domed shaped structure is formed like a small droplet. If this droplet is big, it's a dome like structure. So when this matrigel polymerizes, after that media is added over this dome and over this droplet, and after some time these cells they will divide. After when they divide, they will also interact with each other, and they will secrete the matrix, and after that they will form the small organoid like structure, small these organoids. And these organoids are used for different applications, like they are used to check the efficacy of the drugs, for example drug screening, and other cases. And here in these organoids, tumor cells can also be seeded. For example, this tumor, if here these normal cells are replaced by tumor cells, so these tumor cells can also grow here and they form the tumors. So the morphology of the tumor and other physiological conditions of the tumor, they can be studied from these in vitro conditions. Otherwise we have to study the organs, and we have to sacrifice the animals, or we have to study the tumors directly from the humans. But these organoids will help to study the tumors, to study the to do some these drug screening in the organoids. And these organoids have the as you can see, if consider this as a tissue culture plate, and in tissue culture these cells are only in a one layer. If consider this picture as a tissue culture which I am encircling, so these tissue culture plates, they only grow in a one layer, so the cells do not interact as with they do not interact as much with either. But in case of these organoids or these dooms, the cells are embedded to each other. They have the well interaction with each other, and they form this organoid of cells which have the more cells, and they have the extracellular matrix between, and it gives more advantage for the drug screening as compared to the one layer of the cells. I hope you have understood this. Any question regarding this? If anyone is curious from this in this group to know further what I explain, he or she can ask at any time. So I will move to the next question. If I want to conduct an experiment mimicking moon or space environment, what should I use? Clinostat, weighing balance, thermometer, fluorescence assisted, cell sorter. Yes, most of you have answered correctly. So the clinostat is the instrument where the zero gravity or micro, we cannot say the zero gravity, but micro gravity environment where there is a very less gravity, or the gravity is close to zero, that environment is maintained. So here, this is the clinostat. First, if we want to grow these cells in the clinostat, I will tell you why we need to culture the cells in this clinostat. So these cells are first coated, or they are seeded in a cover slip or some particular type of plate which is used in the different types of clinostat. For example, these clinostats from different companies have different cover slips or different plates for culturing or for growing the cells. So when these cells are fixed, so after that these cover slips or these plates are placed in this chamber, and this chamber is filled with the media. It is completely filled with the media, and after that this chamber is placed containing the media, it's placed in this clinostat, and here it is rotated in such a way so that it simulates the microgravity. Here, when we culture the cells in normal conditions, they are exposed to the gravity, as everything our body, every cell in our body, or the cell culture, whatever the species is, they experience the gravity that's present in the earth. And when we culture the cells, they also experience the same gravity. So in the cell culture, we don't in the normal cell culture, we don't need any of these device to give the specific environment, to give the specific environment to the cells to grow. As our cells in the body, they experience the gravity same way the cell culture plates, they experience the gravity. But in case of clinostat, it is rotated in such a way that the force due to the rotation, it cancels the force of the gravity. So here in this clinostat, inside this, the cells which are present in the clinostat, they do not experience the gravity. So they behave like they are weightless. So why can anyone tell me why we need to grow the cells in the clinostat? What is the purpose? If we are culturing the cells in the clinostat, the, and we are culturing because we need a zero gravity or microgravity conditions to culture these cells. But why we need this? Yes, one reason is plant cells. Another reason, someone is answered to study the plant cells. Yes, it is rotated mechanically. So, as you know that the space scientists, as they go in the space, for example, Mars, Moon, and there are completely different conditions. They do not face the gravity. Gravity is very less. So you may have seen in the movies or in the documentaries or videos that they float there. And the cells of our body, when a person goes into the in the space, like in the earth, the cells have the microenvironment, but they also have the gravity. And earlier we do not know that gravity also has the effect on the cells, but in when the person goes on the space, the cells do not experience the gravity. The cells which are in our body, different blood cells, endothelial cells, cells of our skin, cells of all the organs, they do not experience the gravity. So it has been known that when the person goes in the space, when he or she resides there for 4 to 5 months or 6 months, so the cells, for example, the muscle cells dystrophy occurs. The person becomes weak. These endothelial cells, they become weak. They shed. So this may be because of the gravity, because there is no gravity, or so this may be because there may be some change in the normal physiology because of that. So NASA has usually these cell culture laboratories which have the clinostat. So they study all the things in the disease condition, in the normal conditions, how the cells behave in the space. So they study these all the things in this clinostat, and it will give them idea how the cells will behave in the cell in the space. So they simulate the space conditions of the human physiology in the cell culture lab here in on the earth. So as space does not have the gravity, so we have to simulate here also. We have to create the condition where there is a microgravity, and it gives us the idea when after growing the cells in these microgravity conditions. So after that we will study different pathways which will tell what are the effects on the cells in the when we grow them in the clinostat. So usually the space companies like NASA, they usually have the interest in these type of cultures which uses clinostat. So another question we will move ahead with. So next question is MEMS stands for macro electromechanical system, micro electromechanical system, micro electromagnetic system, and micro electromagnetic macro electromagnetic. What is the answer? Yes, most of you have answered it correctly. The answer is micro electromechanical system. So micro electromagnetic mechanical system. So it is actually the microfluidic device. It it moves the fluids, media, cells from one compartment to another compartment. Or you can also call it is a continuous type of cell culture, but micro. Continuous cell culture. One type of culture is static culture, which is done in the cell culture flasks. So in the flask we add the cells, and after that we add the media, and after some time 24 48 hours when the media is exhausted we replace that. Another type of culture is continuous culture, which is done in bioreactors. So bioreactors, they have the continuous supply of the media. From one side the media goes, and another side the used media, which is spent media, which gets exchanged. So it's a continuous culture. We don't need to change the media. It's continuously going inside and maintaining the normal conditions in the culture. And these micro electromechanical devices, these are also the continuous cultures. In case of cell culture, actually we are talking here. They also have other applications, but here we are talking mostly about the cell culture. So they they are they are used they are continuous type of cultures, but they are very micro in the very small scale. So these media, it is supplied through the small channels which are called micro channels, and these channels supply the media to the chamber which is called cell culture chamber, and in the cell culture chamber the cells are grown, and after that it is this this media, it's taken away by by the micro channels, and it's taken away from these cell culture chamber. So what is the use of this? First I said that it's a continuous, so we do not need to change the media or other things. Another thing is that in this it is a more efficient method to grow the cells. Usually different types of cells are grown, or cells can also be co-cultured in this. So it has been exploited as a organ on chip based systems, in which the, for example, here I have given the example of liver on chip, or heart on chip, or kidney on chip. So in this liver on chip, the hepatocytes are grown, the types of cells and other types of cells which are present in liver. And when the media is supplied, so what advantage it gives as compared to the conventional cultures? In the conventional cultures, only one layer of the cells can be grown, but here in the here are some uh, this in this cell culture chamber there may be some different segments, small micro segments on which cells can be grown on different layers. And as you know that in our body the cells experience some mechanical stress or mechanical force as the cells are in the heart or kidney. They are in the different layers or they are compact, so they also experience the mechanical forces from the other cells as they are compact. But in the cell culture mechanical forces are not given as this, as this is a one layer culture, so cells are not forming a the big bulk in the cell culture plate, in the conventional cell culture plate. So these microfluidic systems, they give the advantage that they have the advantage that mechanical optimal mechanical force can be used so that the normal physiology can be exactly simulated. And also here in this the substances can be introduced as is a microfluidic system. Substance can be introduced and the waste products they can be easily taken from these micro channels, and the waste products or metabolites that are taken from away from these micro channels, they can be studied for different applications or drug screening regularly. And this microfluidic system has, if we want to screen a drug or want to study the effect of drug on the liver, so instead of giving the whole drug to the whole organism to study if it has a potential to treat the liver, we can use the liver on chip, this microfluidic system, where these cells which have the some damage, like which simulates the damaged liver, and these through this microfluidic system, continuous supply of drug can be given, and we can study if they have the potential to treat the disease. Now we will move to the next question. Fluorescence occurs by emission of electrical waves, magnetic waves, electromagnetic waves, none of them. Fluorescence occurs by emission of electrical waves, magnetic waves, electromagnetic waves, none of them. The answer is electromagnetic waves, and most of you answered correctly. Usually in cell culture, different type of dyes are used which have the ability to give the fluorescence signal. Different fluorophores are used. So when the light or photon, when the light of some particular wavelength they are given, and some particular wavelength of the light uh, gets absorbed by these fluorophores. They absorb this light, and due to the absorption of the light, they go from the ground state to the excited state. And and after absorption of the light, as this excited state is the unstable state, they emit the light of different wavelength, and usually this wavelength is higher than the wavelength which they have with which by which they are excited. They have absorbed some energy. After that, the higher wavelength are produced, which has a lower energy as compared to this, as it is the as the wavelength is inversely proportional to energy, and these these electromagnetic waves they are produced from these these dyes, and they are detected by the detector. So those materials which have the ability to emit these electromagnetic waves after the absorption of some energy, so they give the fluorescence. For example, if there is a dye, for example dye A, whatever the is FITC or whatever it is. So this dye, when for example the 500 nanometer light, which is in the blue range, it absorbs this 500 nanometer light, and after that in the excited state it radiates the wavelength of 530 nanometer, which is in the green range, and this 530 nanometer is detected by the detectors, and we can also see the green signal of this as is visible, is in the visible range. And another term which is not related is the phosphorescence. When from the singlet excited state, this dye or substance goes to the triplet excited state, but there is a lag after absorption. In fluorescence, once the this source of the light is stopped, the fluorescence is stopped at the same time. But in case of phosphorescence, this the emission persists for some time. So this is called the phosphorescence. Another question is osmolarity is, what is the definition of osmolarity? Total number of solute particles per kg of solution, total number of solute particles per liter of solution, moles per kg, number of moles. So osmolarity is the total number of solute particles per liter. So can anyone tell me what is osmolality? Molality, LAL. Answer is also in, yes. Answer is in the answers that are given. So you may have heard about the molality and molarity. Molarity is, yes. Osmolarity is the measurement of solute concentration or total number of. Here, why it is written osmolarity? It is the same molarity, but total number of osmotically active solute particles per liter of the solution. So units of this molarity is OSM per liter or osmol per liter. So it is a measure of osmotic pressure of the solution. How solute will diffuse, how solvent will diffuse from uh, through the semi-permeable membrane. So here I have shown the comparison between the molarity and osmolarity. So this osmolarity is a summation of molarity multiplied by the number of moles n. So molarity, as you noted, is a molecules number of moles per liter of solution, but osmolarity is a total concentration of all the osmotically active particles which have the potential or to increase or decrease the osmotic pressure. So on the basis of osmolarity, uh, there are different types of solutions which affect the cell. One is the isotonic solution. Isotonic solution is the solution which has the solute concentration of the cell is same as the solute concentration outside the cell or out in the medium. So these are the isotonic solutions. So water can move from the inside to outside in the both directions. Hypotonic solution is the condition where in the media there is a more osmotically active or osmolarity is high. So the water moves. Sorry, osmolarity is high here in the cell, and here is low. So it is a hypotonic solution. So water moves from hypotonic solution, this culture media to the cell, and this cell lysis will occur. And if the cells are placed in the hypotonic solution, which is the high osmolarity, and the water will come out of the cell and shrinkage of the cell occurs. Another question is osmolarity is measured using electrochemical method or electrochemically, freezing point depression, weighing balance, immunocytochemistry. You may have studied in your, I think, 11th or 12th class that colligative properties, and one of the colligative properties is the osmotic pressure, and osmolarity is actually it's related to the osmotic pressure. So you may have studied that equation where there from this osmolarity. So the answer is the freezing point depression. So from the depression of freezing point, this osmolarity can be measured, and this freezing point depression, when the solutes are added in the solvent, and because of the solutes, the freezing point depresses, freezing point decreases. So the equation for this is change in the freezing point or change in the temperature is equal to temperature of the solvent minus temperature of the solution, as the solution has a less temperature as compared to the only solvent. Another question, which is the last question, cell attachment can be promoted by treating the substrate with. The answer is cold trypsin, warm trypsin, polylysine, antibiotic. So most of you answered correctly. The answer is polylysine. So can anyone tell me the use of the trypsin in the cell culture? Yes, Rajesh. Anyone else? So Rajesh have answered correctly that trypsin is used to detach the cells. Yes, it digests the Nandini have given the yeah, this exact answer. It digests to the extracellular matrix, and because of the digestion of extracellular matrix, the cells they detach from the cell culture plate. And besides this poly L-lysine, nowadays it is mostly used in the cell culture plates. Earlier, what substance was used? I think I have given the answer in earlier sessions. Besides poly L-lysine, so in the 90s, yes, Rajesh has correctly answered collagen. In the 90s, before the discovery of this polyalkyl lysine can help in adhering attachment of the cells. So earlier, the when the cells are grown on, these animal cells are grown in the cell culture at the first time. So these cells they were not able the encourage dependent cells which attach to the flask, they were not able to attach the plastic or this glass, as the plastic they have the hydrophobic surfaces and cells only attach to the hydrophilic surface. So firstly, usually the plasma was coated earlier. The plasma from the blood, it was coated in the cell culture plates. But this plasma, it has a disadvantage that it cannot adhere properly in the flask. So there were not the optimum this cell attachment. After that, extracellular matrix like collagen was used in the late 90s. So collagen was used as a coating material for this. And now this polylysine is is mostly used as it is a non-specific cell surface attachment. It does not have a specificity to any type of cell. And each and by what mechanism the cells are attached to this? Cells attached due to the electrostatic interaction. Due to the there are negative charge there in the cell culture membrane, which interact with this polylysine, and they will adhere to. And the cell culture flasks which are nowadays which are supplied, they are mostly made of plastic, polystyrene. This polystyrene has a hydrophobic environment and the cells will not adhere to this polystyrene. So this polystyrene are coated with polylysine, and polylysine gives the optimal hydrophilic environment, and they also enhance the cell attachment to this electrostatic interaction. And this polylysine is also the non-specific, so any type of cell can adhere to it. So they were the questions of week 3, and this is the YouTube channel on which I will upload this recording of this video. If anyone wants to watch it later, or there are also other video lectures or which I have uploaded in, so you can visit this channel and you can watch the videos. If anyone have any questions. Thank you, Lemi. So if you have any other questions, you can ask. Otherwise, I will conclude this session. No, no, magnification power will not affect the light. So the light is actually adjusted. When there is a light source, it is adjusted. Magnification also only helps you to magnify. It only has some magnification lenses. So it only contains the lenses. Light source is different from the magnification lens. So the light, there in the microscope, there is a separate knob for to control the light, and the light does not affect the magnification. It only helps you to visualize it properly. If if there is the optimum light, you will be able to visualize the cells properly. But if there is a more light, so maybe the your cells will be something like hazy because there is a more light. It will they will blur with the background. So thank you so much. Thank you everyone for joining this session. Hope to see you in large numbers in the next session. And you can also interact if uh, you feel whatever I talk. If you feel to ask question, or if you are curious to know more about this, and or if you have any confusion when I explain any question or slide, so you can ask. You can unmute yourself. You can ask. So thank you so much. I will now, if there are no questions, I will conclude the session. Thank you so much. See you next week.